Thanks, Yuichiro. And I want to extend my thanks again to Eric and the Scientific Organizing Committee for putting together a great series of talks. And I'm going to try to hook my talk in here from a perspective of trying to understand ways that we can observe the historical record of catalytic evolution through time. And this is a very broad question. And I'm going to use a case study of looking at microbial sulfate reduction as a system to try to see if we can do this, if we can start to link enzyme sequence evolution to fractionation of stable isotopes and be able to monitor the historical record of those isotope fractionations through time. But I'm also interested in doing this from the perspective of understanding cells today, from a microbial ecology perspective, where we might be able to use stable isotope fractionations as a way of gaining insight onto the physiological state of a cell that we might have in a bottle or have in a lake or some other environment. So this is I'm presenting today, but a lot of the work was done by Min Sub Sib, who was a postdoc previously in my previous affiliation at Caltech. He's now at Seoul National University. And here at ELSI, Chris Butch and Mackenzie Smith are also working towards some of the goals of this project. And so, I titled my talk, and I'm really going to center the talk about this idea of using stable isotopes to understand cellular physiology. But I want to make it a little bit more general than that, and hopefully be able to get some criticism and comments and other ideas about how we might be able to use this approach of looking at stable isotope fractionation coupled with knowledge of catalytic kinetics to be able to follow enzyme evolution through time. And so broadly speaking, we have two main repositories of the knowledge of biology on the planet. One is in stuff that we can go out and see, like these piles of cells that we call stromatolites, which appear fairly early on the Earth. And we also have other material repositories, these stable isotopes. And so light carbon, like as shown here, this is a reference to one of Yuichiro's papers, that there's a sign of metabolism deep in the past that's apparently recorded by the distribution or fractionation of light and heavy carbon, the different isotopes. So other than these material phases that we can go out and find in old rocks, we also have molecular biology. And we've seen a couple of phylogenetic trees come up in the last couple of days, and a couple mentions of the concept of using molecular clocks to try to put a timeline on how DNA and protein sequences evolve through time. So these are kind of two main repositories of biological knowledge. And what I'm going to propose is a pathway to be able to link these two, to link sequence evolution of enzyme catalysis to actual repositories. And I think that might help us calibrate things. This is a long-term goal that I have here. So the model system that I'm going to discuss today is microbial sulfate reduction. This way of thinking about things is not limited at all to microbial sulfate reduction. But microbial sulfate reduction is a convenient starter system for this way of thinking, because there is a long history of measuring stable sulfur isotopes and their patterns of fractionation throughout geological history. Microbial sulfate reduction is an 8-electron addition onto sulfate to make sulfide. Sulfate is a pretty oxidizing molecule. And so what these organisms are doing are taking electrons and using the energy released when those electrons move on to sulfate to power their growth metabolism. And they're fairly diverse too, I should say. This is the net equation that we could write for this process. We could calculate the energy of it, think about the cellular yields, but they're a quite diverse group of organisms that are all breathing or respiring sulfate. And as I said, there's a long tradition in geochemistry of measuring stable isotopes of sulfur through time and also in different environments. And so what I'm showing here is a compilation of sulfur isotopes between sulfate and sulfide. And we're looking at the two isotopes of sulfate of sulfur 32 and sulfur 34. And what is being shown here is in this gray area, the equilibrium fractionation values of sulfate to sulfide for 0 degrees Celsius all the way up to 40 degrees Celsius. So the temperature at which sulfur isotopes will equilibrate between sulfate and sulfide will affect the final value of the system. And then we have all of this other stuff out here that I labeled kinetic. This is the stuff that life does. And so life is able to operate its metabolism in a way that's fast enough that sulfide is produced so fast that the isotopes aren't able to equilibrate. And so we get these deviations from equilibrated sulfur isotopes. And these kinetic fractionation factors have been thought to be related to all sorts of stuff. The rate of the organism's growth, the place that they grow, are they marine, are they terrestrial, are they pure culture, are they mixed culture? And it still remains an issue to define exactly which parameters are affecting the kinetic isotope fractionation values that life, the result of biology, and why and which magnitude these isotopes are deviating away from the equilibrium. If we spread those data along an axis of time, and we think about long ago here and today here, and we look at sulfide isotopes compared to sulfate isotopes, sulfide in the form of pyrite that's preserved, we can see that in modern day environments there's a huge spread of sulfate-sulfide isotope fractionation. And then in the past, we have this problem that there's not a lot of data because there's not a lot of old material on the Earth, but the kind of variance here collapses. And what I've marked on here is this 20 per mil fractionation value between sulfate and sulfide. And so I'm going to try to address the question or speculate on why this 20% difference might be in the Archean, but in the present day environment, we get these very, very large fractionations in biology. So what's going on in this process of sulfate reduction? Sulfate, I mentioned before, it's a fairly oxidizing molecule. You can put electrons onto it, and when energy is released as electrons go onto sulfate, gain energy from that process, but it's actually chemically inert. And what organisms have to do is they have to activate sulfate by using an ATP, and in this activated form, they're able to put first 2 electrons onto sulfate to make sulfite, and then there's a 6 electron reduction of sulfite to make sulfide. So this is the pathway, or a very simplified version of microbial sulfate reduction. The process of breathing on sulfate, step 1 is to overcome this kinetic barrier and kind of power through that by using an ATP. And the second step is a first 2 electron reduction, and the third step is this pretty awesome, I think, 6 electron reduction of sulfite. And for a long time, people have recognized that there's a pretty neat relationship between the ability or the extent of fractionation of sulfate-sulfide isotopes proportional to the rate that these cells are growing. So let's explore this diagram a little bit. It's a simple curve, but it's a little bit tricky to think about. I said before, let me go back, that the equilibrium value is essentially set by temperature over here. It's these high values over here. This is around 60 per mil, 80 per mil here. And what biology is doing is spreading the data away from that equilibrium fractionation. This is another way of drawing that, but it's a way of drawing it that emphasizes a rate dependence on the process. Here we're talking about fractionation factors in an inverse way, so this is a little bit confusing, but we could translate this number here to about a 60 per mil fractionation. That's that equilibrium value that would be met as the rate goes to 0. We would encounter the y-axis here, and that would happen, depending on the temperature, right around 60 to 80 per mil fractionation. But as cells grow faster and faster, the fractionation changes. It's kind of muted away from that equilibrium value. So this is an old observation that has been reproduced again and again, historically about every 10 years, actually. Another group is able to reproduce this curve, and it leads to this question of, like, why does this happen? And a couple years ago, I think there was a pretty nice breakthrough in my mind, with trying to understand the shape of this curve. And that breakthrough was accomplished in a model that related the rate of the process, the energy of the process, and knowledge of the fractionation factors of these enzymes, or the inferred fractionation factors of these enzymes. And really, this is coming out of some work a while ago by Clinton Stoner, and which was later on followed up by Dan Beard and Hong Kim. I don't know how to say that name. I shouldn't try. Sorry. These folks of trying to relate reversibility of a chemical process to isotope backflux and the force or the chemical potential of a given reaction. And so this knowledge of being able to relate the chemical potential of a reaction to the backflux, which is related to isotope exchange, was employed by Boswing and Yamei Li in a 2014 paper where they were able to put a line on this data by building a model. And so they could say, okay, now we have a model that describes the state of a cell. We know the parameters of the model, and we can make a line that actually fits the data. And so this is very, very satisfying because what they were able to do is infer the processes that are occurring inside of a cell. Here's a cell that's doing microbial sulfate reduction, and they broke the process apart and put those components into their model, which is relying on knowledge of rate and isotope fractionation to say when sulfate is imported into the cell, there might be a fractionation that occurs. And then here's our activation step. This is to get rid of this kinetic problem. Here's this third step, the first 2-electron reduction of the sulfur atom. And then there's this final step, the 6-electron reduction. So this seemed like a satisfying way of looking at things to be able to understand the shape of that curve. But it left us still with a little bit of some missing data. We could still take the model and come up with different scenarios to accomplish the same isotope fractionation. This is a common process in science. Many hypotheses can account for the same data set. And so there's a degeneracy built into this model. And that degeneracy I'm seeking to get rid of by trying to find out two pieces of knowledge that have not really been described. One, what are the actual fractionation factors that are happening at the different steps in the metabolism? These have never been determined empirically, and instead they've just been estimated numerically. And also, what is the rate determining step of the metabolism? Knowledge of the rate determining step of metabolism is very, very important in this case because that's going to be the bottleneck that will be expressed. That'll be the fractionation factor that we actually measure later on. It will be dependent on that rate determining step. So to try to determine what that rate limiting step is, or in part, to do that, MinSub was actually able to make some measurements of intracellular concentrations of these sulfur metabolites in the cell. And here's a complicated graph, and MinSub uses kind of funny symbols, so be careful here. Here is cell growth. Here we're monitoring the production of sulfide. It's the respiratory product of the cell. So the cells grow, sulfate goes down. And sulfate inside of the cell kind of bounces around in concentration. It's not clear really what it's related to up here, but it's pretty high in the cell. Then here's the concentration of APS in the cell. This is the kinetically activated sulfur compound that those first 2 electrons will be put onto. And then here's the sulfite. This is the compound that has 6 electrons added onto it. And what MinSub found was that basically the APS is always at a higher concentration than sulfite. And so one thing this implies is that kinetically this APS reduction step is a little bit slower than the sulfite step. And once sulfite is produced, the final 6 electrons go right onto sulfite, making sulfide. And this seems fairly comfortable chemically. Sulfite's pretty reactive. And so it kind of goes to this suggestion that maybe this APS step is rate limiting. And if that's the case, this APS step will be very important in controlling the observed sulfur isotope fractionation factor of the metabolism. And so we could say that possibly this rate determining step, or at least in some cellular conditions, would be that first 2 electron reduction of APS. And that leaves us with a second part, missing part of the data here is the actual numbers that are associated with these enzymes. as an individual catalyst, what is the stable isotope fractionation of this catalyst? So we sought to address that question by purifying the enzyme and assaying it using isotope ratio mass spectrometry of the substrates and products to determine that value. So here's the enzyme. Remember, it takes this kinetically activated form of sulfate and adds 2 electrons onto it to make sulfite. Here are some other iron-sulfur clusters that are found in biology. This time, these iron-sulfur clusters are relatively high potential. They're not sitting at minus 500 millivolts, like a ferredoxin. They're more oxidizing than that, and that's because sulfate is more oxidizing than that. You could imagine electrons coming in to these 4 iron, 4 sulfur cubanes and then being transferred on to this FAD, which is hiding up here in yellow. And then at that stage, sulfite is going to dock somewhere around here. Or sorry, the APS is going to dock around here, and sulfite will be produced. So Hideaki Ogata, who was previously at the MPI for chemical energy conversion, he grows 200-liter vats of this organism, Disulfid Vibrio vulgaris. And so he was able to donate some of this protein material to the laboratory and save us a lot of work. He did a partial purification of the APRA and B subunits and sent them to us, at which time MinSub was able to conduct protein assays. And what he did was he put the substrate of the enzyme, and an artificial electron donor into a tube in an anaerobic environment, and then he was able to use ion chromatography to separate out the products and the substrate of this reaction. So in this case, he's going to collect APS, the substrate of the reaction, and sulfite, the product of the reaction, and then later on take those molecules and launch them into the Neptune instrument, and we'll be able to determine the 32, 34 isotope values in a compound-specific way and be able to understand the isotope fractionation value of the enzyme. Here are the enzyme kinetics at 2 different temperatures. And so what MinSub was able to show was that cold slows the enzyme down and heat speeds it up. So a change of 12 degrees changed the rate of sulfite production by around 5 times. So a pretty dramatic change in the rate of the enzyme. And he was able to monitor the sulfur isotope compound value for APS, the substrate of the reaction, and the product of the reaction, and then make these basically Rayleigh fractionation curves where the slope of this line is indicative of the 34-32 sulfur isotope difference between the product and the reactant here. I said before that the rate of the enzyme here is 5 times faster than the cool 20-degree assay version here, but something interesting, the isotope fractionation factor is basically the same between these 2 cases. And so with this in hand, we have a model that describes mathematically how isotopes will be partitioned from sulfate to sulfide under various cellular states. And now we have a real number now, the first number of a purified enzyme. I should qualify that statement by saying entirely pure, non-missing subunit version of the enzyme here. Previously, about 2 years ago, another group measured an enzyme, but it was missing a subunit. And so this was actually the first purified, complete enzyme component for which sulfur isotope values have been measured. And we also have this data before, which is suggestive of this rate-limiting step of this enzyme here. And so what we did is we took this previously constructed model and we started plotting expected isotope fractionation values of the cells for different chemical conditions in terms of the driving force of the metabolic reaction. So if the cells have lots of energy, that would be sitting over here at these more negative redox potentials. And if the cells have less energy, they would be using more oxidizing electron donors or higher potential electron donors to reduce sulfate, and they would have less driving force behind their reaction. And what we found when we started plotting the data like this in this model was that actually the observed isotope fractionation of the cell does not ever go over the isotope fractionation of this one single enzyme until that enzyme step becomes reversible. And so this is this backflux contribution here. When there's not a sufficient amount of energy to push that reaction forward, the isotopes can re-equilibrate across between APS and sulfite, and that's what leads to these large, close to equilibrium fractionations. And we were able to do that for a number of different rates of microbial sulfate reduction. And you can see when cells are really cranking through sulfate and making sulfide, that kind of mutes everything. They're pulling the sulfate through their metabolism so fast that it doesn't ever have time to approach these equilibrium values. But at these slower rates, again, the situation, as long as the cells have sufficient electron driving force, the sulfur isotopes can't really ever go above this value, which is set kinetically on this one particular enzyme. So that's just what I said. It can't increase more than the actual APS enzyme step when there's sufficient energy. So that's kind of the nuts and bolts of the data that I want to present, and I want to kind of hypothesize about the possible implications of this purified enzyme sulfur isotope value as it might have something to do with our historical interpretation of these isotope values. And so we can ask this question, why in the Archean do we not see close to equilibrium sulfur isotope fractionation values? Here we see these really, this really big spread. Some of them are narrow, but there's a lot of variance here. And one thing we might say is that in the modern environment, there's a huge diversity of environments, and in particular, these organisms are under a lot of competition, and they might be using very modest electron donors. And those very modest electron donors put us over here on the right side of our graph, where the cells are growing closer to equilibrium and therefore relating these larger isotope fractionation values. It's very tempting to speculate that, using a similar line of thinking, that in the Archean, the fact that we don't see any large sulfur isotope fractionations might be coincident with the idea that these organisms were electron replete. They had a lot of power to run their metabolism, and they essentially weren't starving for electrons. And to help us kind of probe around this idea and interpret this idea, we can think of what sulfate reduction might have meant evolutionarily if you were a microbe swimming in the ocean in the Archean and you were using hydrogen. If you were a sulfate reducer or a methanogen or an acetogen, we could use the concept of the threshold concentration of hydrogen and the expected energy yield for the same concentration of hydrogen to either take electrons from hydrogen and reduce sulfate, or take electrons from hydrogen and reduce CO2 in either of these two different ways. And because sulfate is such a good electron acceptor, because it has a more positive midpoint potential, it's easier to reduce than CO2. What sulfate reducers are able to do, and people have known about this for a long time, they're able to suck hydrogen concentrations down to a much lower level than either of these organisms. I think a good way to interpret this is just to look at these Gibbs free energy of reactions for the electron addition onto either of these acceptors. And so probably the evolution of sulfate reduction was a major bioenergetic innovation, and what that allowed these organisms to do is, in the case of using a common electron donor, was they were able to outcompete anything else that was living around there, putting electrons onto very meager electron acceptors like CO2. And that would be kind of coincident with this idea that they were running electron replete metabolisms. Today, these organisms are shoved into all sorts of different energy environments, and they're living off of a lot of scraps, where they don't have a lot of driving force in their reaction. And that might be one way to kind of interpret this historical distribution of stable isotopes through time. Very replete conditions, lots of electron donor. They're competing against things with very modest electron acceptors. But today there's a lot more competition. There's a lot more high potential metabolisms available. And I want to take this concept a little bit further and use our knowledge of isotopes here to speculate on processes of energy conservation that might be occurring in these cells. I said earlier that these microbes are quite metabolically diverse, and they are, and in fact, we don't have a unified understanding of how electron transfer processes are coupled to energy conservation in these cells, or energy conversion, if you will. To say that simply, we don't know which steps in these metabolisms are coupled to chemiosmotic potential generation. And that will probably be variable for different types of cells, but it's interesting for me to consider whether or not we could use isotopes as a way of making hypotheses that are testable here. So here's a picture of a cell that might be putting electrons onto sulfate and pumping ions out in that process and then making ATP as those ions come back in across the potential. And we could think about growing a sulfate reducer in a laboratory environment. This is something that I would like to do, or if you want to do it, please talk to me and we can do it together. If you have some sulfate reducers growing at home. But you could imagine growing sulfate reducers with a chemical potential for their metabolism, and you could set them by modulating the concentrations and the temperature, and you could grow them at this potential, and you could say, well, if this organism was pushing against a membrane-bound ion potential of 180 millivolts, that would translate into about 20 kilojoules per mole, and then you would expect the cells to be able to gain or to have a free or remaining amount of energy of about 10 kilojoules per mole of reaction. This would mean more reversible and more fractionation if it was coupled. But if you took those same cells and they weren't coupled, they weren't pushing against the ion potential, they weren't using this to directly conserve energy, you would get 0 minus 30, so minus 30 kilojoules per reaction. And from a sulfur isotope perspective, what this would mean would be a condition where it's less reversible and less fractionation. So this is a testable hypothesis that we might be able to use isotope fractionation from whole cell environments to be able to find something out about their biophysical energy transduction system. So the next steps that we're pretty excited to do now that we have one data point on one of these enzymes is to try to entertain this possibility of looking across the sequence phylogeny of these sequences that are found in different organisms, these different homologs, and to see if there's any evolutionary variation on this catalyst through time. And I think that will be very, very important for us to understand whether or not this hypothesis that I set up it could be true at all. If there's a lot of isotope variability, certainly we can't go and make claims about how this minus 20 per mil number has meaning in deep time. Instead, we'll be left with a question of what are the ancestral state variants of these enzymes operating at in terms of their sulfur isotopes. And so that's something that we'll do in the future, is try to gain some evolutionary insight onto the history of sulfur isotope fractionation from the perspective of individual protein sequences. And I think this will be a really nice way of us to try to bridge these two biological records that we have on the planet. We have these material records, such as the distribution of isotopes through time, and we also have this molecular biology record. And I think these isotopes might be a nice way to connect these two pieces of information together. And the next place we're going with this, in addition to that, is to try to understand enzyme mechanism from these stable isotope fractionations. This enzyme has a pretty cool proposed intermediate. This is one of those intermediates in the proposed catalytic cycle, and what you can see here is a FAD up here. Those 4 iron 4 sulfur clusters would be kind of off the stage, floating up here. Electrons come down into the FAD, and APS is reduced, leaving a covalently linked sulfite adduct here. So this is a pretty interesting proposed mechanism that hasn't received sufficient testing, I don't think. But one thing we're trying to do is use this measured isotope value to test this proposed mechanism. And I think that might be possible because these KIEs are kind of reports of the bond vibrations that can be expected in this type of environment. So we might be able to approach that from a computational perspective. In the beginning of the talk, I said that this type of approach, even though I'm going to talk about sulfate reduction, it's very applicable to other microbial metabolisms. We could look at some of Kameya-san's enzymes with a similar approach to look at the isotope fractionation of carboxylation or decarboxylation reactions. We could also find interesting knowledge from testing other metabolisms, like these apparently reverse metabolisms that I've put on here, where one organism is putting electrons onto CO2 to make methane, and another one is taking electrons off of methane to make CO2. So I hope that this way of determining enzyme-specific apparent kinetic isotope fractionation factors will help us understand a lot of metabolisms and how sequences evolve through time catalytically. And then, when I was watching George's talk, it occurred to me, really in force that if we're careful, we will also be able to look at kind of these proto-metabolic networks. And if we can start measuring catalytic or rates of these types of reactions by different catalysts, we'll be able to start to use these types of isotope measurements to understand in more detail these non-biological reaction networks. And so that's where I'm going to stop with my talk, and I'm going to open it up for questions there. And I just want to mention that this last year, me, Batul Kachar, Daniel Segre, Boz Wing, Chris Butch, and Chris House, we started a proposal to look at ancient thioester chemistry from a number of different perspectives. One of them involves looking at promiscuity of thioester utilizing enzymes in biology. Haruyuki Atomi gave a really, really fascinating talk a couple of days ago when he was talking, and he mentioned that some enzymes are able to use N-acetyl-2 amino ethane thiol, I got it, instead of acetyl-CoA as a cofactor, and I think that is pretty interesting from an evolutionary perspective. But we're also interested in making carbon and sulfur isotope measurements of these enzymes and trying to find out, in a similar vein as what I presented here today, how these isotope values may change through time or not. And we're also interested in trying to constrain how much thioester metabolism might be possible in a metabolic network today or in the past. And so with that, I'd like to thank everybody for your attention and give a special extra acknowledgement to the funding that was supporting this process, this progress, and which was provided by NASA, and also more recently from the WPI program here at ELSI. So thank you very much.
Okay, now the time for question.
So, coming back to one of the themes of the meeting, that there's always a lot of chemical stuff going on, but maybe some of it matters and some of it is just kind of noise along the side. The statement, this is a question about whether this is a correct way to look at things. Your statement that sulfate is a relatively unreactive species, and so you have to do something unusual to it to activate it, reminds me of what is the case also with nitrate, which has tremendous capacity to oxidize, but it's difficult to get at. And it's a little bit like the Rubisco problem, that CO2 is not particularly reactive, and it's not all that different from oxygen. So activating it and distinguishing it, discriminating it is hard. All of the enzymes that do this seem to have big isotope signatures, and the signatures seem to be related to how hard you can drive the reaction. So we see things like a regular relation between isotope fractionation and the discriminating capacity in Rubiscos and things like that. But the great thing about these non-reactive species is that they should be the chemical bottlenecks that wind up trapping free energy in one domain without a lot of reaction partners for it, so that it can transport globally and become sort of the major carrier of disequilibrium. Should we expect that the major isotope signatures are the way they are because the major opportunities for biochemistry to take advantage of kinetic traps are these molecular, like, following Everett's point, are these molecular species that are hard to activate, so the enzymes that can figure out how to do that are frequently going to wind up showing these kind of signatures?
I'm not sure. I think that's a really great question. I think there are probably examples of enzymes that are doing overcoming, well, nitrogenase is one. It's overcoming a huge kinetic barrier, but its isotope fractionation is really small. And so it's hard for me right now to understand exactly how what's general there. Yeah.
So that was actually the question I was just going to ask you about nitrogenase, but I've got a second one. You showed this huge spread of, it wasn't your data, but a huge spread of isotopic fractionations, and people have tried to separate them out based on environment type and so on and so forth. But has somebody, I mean, maybe this is obvious to people that do this work more, but complete versus incomplete oxidizers, whether or not they use cytochromes versus disulfiram, different major physiological variations in sulfate reduction that I believe are pretty well prescribed onto the evolution of like the sulfate reducers themselves. Can you start backing out some of these patterns of fractionation based on cellular physiology and whether or not that tracks molecularly in the evolution of these taxa?
I think that's a really great idea. I'm not aware of anybody doing that in a comprehensive way. That Harrison and Thode paper from 1957 has guided the field really strongly to try to relate cell-specific sulfate reduction rates to the observed fractionation. And so people have gone out and measured many, many types of SRB and only parameterized it against that rate. But I think from what we know now, especially from that model that Boz and Itai constructed, and then our and their also recent permutation of that, of being able to spread the data along an axis that is the amount of energy released in the reaction, we should be able to do just that. I think it's a really good idea.
So, okay. Yep.
Thank you for your talk. It's very interesting. So you have planned to apply the similar strategy to study the carbon isotope fractionation. So in your case, you will study very pure system, like one enzyme or one organism. And what's the molecules that you will try to target? Like carbon dioxide, methane, or some like this? It's a mineral record. Which data you will to
right? Yeah. So
compare?
There's a couple. Yeah, there's a lot of different. It depends on the question that wants to be addressed. I think the autotrophic ones will be really interesting. For example, acetyl-CoA synthetase would be a really interesting one to look at. But in the cell, even for non-autotrophic pathways, I think if we can,
so
we have to know what the rate limiting step is in a pathway, and then that helps us understand which one is the important one to look at. And then for that question, getting that enzyme-specific number.
Okay, so the question will not be to explain the mineral record, but to understand a relatively pure system in biology.
Yeah.
Okay,
yeah.
Understand.
No, it's okay. All right, so if not, so thank you very much.